All right. All right. So I wanted to pick up where we left off in class. And again, this will be we're going to cover basically the remainder of the immune lecture because now we've kind of set forward all the different components of this. And so we've talked a lot about the innate system and the innate effectors and all of these things. And in class this past week, we had discussed this concept that when dendritic cells travel to the lymph nodes, right, they are activating T cells because T cells will eventually give rise to the B cells, and we'll talk about how that all works in a few moments. So again, macrophages and dendritic cells, they can express um, they can express MHC class 1 and MHC class 2. And MHC class 1 and MHC class 2 can be important or are important for the ability to activate certain subsets or certain subpopulations of T cells. The two different kinds that we have are CD4 positive T cells, which are our T helper cells, and as they are T helper cells, their predominant role is helping the immune system it by way of secreting cytokines. And that secretion of cytokines plays an important role in activating CD8 positive T cells, the other type of subtype of T cells, as well as enhancing other innate immune cells and their ability to function. And CD4 positive T cells are activated when MHC class 2, so MHC class 2 is expressed on these antigen presenting cells. When MHC class 1 is presented, the MHC class 1 molecule is received, it is only able to be recognized by T cells that bear the CD8 marker. And this is because CD8 and CD4 and CD8 are two biomarkers, they're two receptors, which are co-receptors for these certain kinds of MHC molecules. And so in T cell biology, when you're born with an organ called the thymus. And the thymus produces T cells. And how this works is that as you get older, T cells and B cells, and just so we can kind of moniker this, B cells are made in the bone marrow. So just so we can, get through all this, so the thymus creates T cells, and as it produces T cells, it's producing T cells with different recognition components. So every different T cell that you have in your body can recognize a certain kind of epitope of a certain kind of antigen of a certain kind of pathogen. And so what you end up getting is this very high level of specificity, but that specificity in T cells and B cells is pre-built in, meaning that this occurs kind of at random through the generation of the molecules that recognize presented antigens in the MHC molecule. And those molecules are known as the T cell receptor and the B cell receptor, which is going to give rise to what B cells secrete as antibodies. So this is what an antibody or B cell receptor look like here. And this is a T cell receptor. And what you can gauge from this is that these have two very common regions in that they both have a variable region, and that variable region is a facing to the outside component of these molecules. And the variable region, or the Fab region, the fragment of antigen binding region, is going to be what is going to recognize the molecule. So if we have an epitope here and it is bound by an MHC molecule like so, T cell receptor and B cell receptor can recognize it like this. It has a component that can recognize both, or that fits into both the clefts of the MHC molecule and a cleft that recognizes the epitope of the antigen. So these regions are called variable because B cells and T cells, each B cell in your body and T cell in your body, they can recognize different epitopes, different amino acid chains that are presented on by antigens. And so that's why they have to have a component of their protein structure, their protein complex, which is variable, so that one can recognize one thing and a different one can recognize something different. There's also a region called the constant region, and these are more cytoplasmic or more outward facing, or facing away from the binding site of these molecules, and they exist to for two different reasons. Two different reasons. The first reason for T cells is that the constant region is what binds the T cell receptor into the membrane and allows for subsequent signal transduction in order to activate the T cell. For B cells, the antibody production is actually, if you think about it like this, from this perspective, if this is my antigen, and my antibody recognizes this region here, the antibody, the part of the antibody, the variable region, would need to bind like this. And so therefore the constant region actually effaces out. It actually effaces out to out into the environment. And if you think about this, antibodies kind of act like C3b in complement, in that there is a component that sticks out, and so there's ability to bind to an antigen, to a pathogen. There's a component that sticks out of this molecule. So if you're thinking to yourself, well, if there's a component that's constant, right, that means that it's non-variable. It has very short, very short medley of variability. Then there should be a receptor on another type of cell that can recognize it and engage in to allow the antibody to engage in opsonization, increasing phagocytosis. Now, the interesting thing here is how exactly we get this type of variability. And without getting into too much detail, because it's a little bit beyond the scope of what this lecture is about, the variable region for antibodies and B cell receptors, as well as the variable region of the T cell receptor, exists in a way that exists in a locus called the VDJ locus of the immunoglobulin family. So the kind of molecules or protein complexes that we talk about are called immunoglobulins. That's the major subclass of proteins and protein complexes that these belong to. And the variable region is created by a process known as VDJ recombination. And this basically is that you have a segment of different genetic loci called the V region, the D region, and the J region. And in these regions, you have a large series of different loci, and they have in between them interspersed sequences which are slightly homologous. And so what ends up happening is you end up getting a connection between one component of the J, one component of the D, and one component of the V region, and they then combine to make one new variable binding site. And then in a different uh, type of recombination, you take another component of V, combine it to another component of D, which combines it to another component of J. And so these different regions use their areas of homology to undergo recombination events and then merge a brand new locus or brand new gene product that creates the variability within these molecules. So it is the binding of the T cell receptor. It's the ability for the T cell receptor to bind to sorry, 1 second. It's the ability of the T cell receptor and the antigen binding site of the T cell receptor. When this happens, you end up getting activation of your T cells. So you have a T cell, the T cell receptor exists like such. So this is the TCR. And when it can bind, when it can bind an epitope bound to an MHC molecule, so we have an epitope here, we have the MHC molecule here, and this is bound to a dendritic cell. This interaction or this engagement allows for a signal transduction which turns on the T cell. And again, if it's CD4 positive T cell, it will become a T helper cell. And if it's CD8 bearing, because it's binding to MHC class 1, then this will allow for cytotoxic T cells. And again, if we think about this, right, any type of tissue that you have, any type of tissue in your body can become tumorigenic, right? So there has to be an ability to recognize endogenous antigens, just like when you have a viral infection, all of those antigens are going to be expressed on MHC class 1 because they're endogenous. Because again, viruses are obligate endoparasites. So if you think about it from the logic, right, if a cell is infected with a virus, right, it's going to express MHC class 1 to tell the world that it is infected with said virus or that it's become tumorigenic. And so the logic is that if the dendritic cell is expressing MHC class 1 and triggering the activation of cytotoxic T cells, again, have the ability to recognize specific MHC class 1 loaded with certain antigens and have the capacity to kill that cell in order to make sure that the virus or the tumor does not continuously spread. Now, it's also one more important thing to note that, these T cells have, again, the same granules that we talked about with natural killer cells. So granulysin, granzyme B, and peripherin. And they generate the same general process in that when the T cell interacts with this is death by apoptosis. Okay. Sorry. All right. So carrying on. So now with B cells, B cell receptor again has the ability to recognize um, different epitopes and different antigens. And one other thing I'll just quickly see if I do this right this time. Sorry. One thing else I want to just get through is what exactly is a T cell actually recognizing, or what we're about to see with B cells. Well, if we have a bacteria, and that bacteria, and again, this is an example, has a protein on it. This protein, when it's broken down by the processes of phagocytosis and then proteolysis, is going to have different components. So let's say that the protein, if we were to break open the protein and kind of string it out, it looked something like this. So it looked something like this, just as an example. When this protein is destroyed by say a macrophage, what is loaded on to the MHC class 1 molecule is very much a component of all of these things. So it's very possible that the macrophage looks like something like this, and this is the macrophage, has loaded all three components as epitopes like so. And this can potentially activate three different kinds of T cells, potentially. And so these three T cells, or we're about to talk about B cells, can all find this different components of this particular protein, which exists in this bacteria. So there's a potential that you could have 15, 20, 30, 100 different subtypes of T cells, which can see that particular bacteria or that can see that particular virus, because they're responding to, or their selection, their ability to recognize these things is in the ability to recognize an epitope, which is a component of a component of a certain microorganism. I think I gotta do it this way. All right, so with B cells, on the other hand, B cells are not activated by way of MHC recognition, because again, dendritic cell, right? So dendritic cell, right? So DC, loading, expressing, activating T cells first. But as you might remember, right, when I talked to you guys last week, the dendritic cells are migrating into towards the afferent lymph and through the afferent lymph to the common lymph node. But also, because of this increase of fluid expression and drainage and increased vascular permeability, we're getting flushing or movement of whole microorganisms down into the draining lymph as well, down the afferent lymph node. This is a critical component of how B cells are activated, because B cells, again, they're going to have B cell receptor, right? So this is B cell receptor with the same antigen binding site that occurs here. And that antigen binding site on B cell receptor is going to engage with those particular antigens that they recognize. And what ends up happening is, I'll draw your attention up here. What ends up happening is, if this is the antigen that the B cell recognizes, the B cell receptor can be engaged. And once it's engaged and both arms are engaged by that particular antigen, it causes phagocytosis of that antigen and eventual proteolysis of that antigen and loading onto MHC class 2. Because again, and this is a really good example of this is exogenous, right? It's seen on the outside of the cell. And if you want to think about it from the logical perspective, if we've covered viruses and tumors, and all we have left are your multicellular parasites and bacteria, fungi, and protists, all of those microorganisms can be extracellular, they can be intracellular, but they have to have an extracellular existence prior to intracellularization, if they are intracellular. So at some component of their life cycle within the human host, those microorganisms have to reemerge. Whereas a virus can very happily, infect a cell and just hang out in that cell for years, even a whole lifetime. So that's why cells, or we have evolved to recognize things that live, that have a component, that have ability to have a component of their life cycle where they are extracellularized, that we then have a way to enhance our ability to recognize those particular microorganisms. And so B cells are phagocytes, and again, they are one of the 3 antigen presenting cells, right? So as professional antigen presenting cells, once they phagocytize those antigens or whole microorganisms, they destroy them, load all the epitopes onto MHC class 2. These are then brought to the surface, and then T cells, a very specialized kind of T cell called a T follicular helper cell, because they live in the follicles of the lymph nodes, have a T cell receptor that can recognize this new MHC molecule with its particular epitope. This triggers the release of cytokines, because again, they're a T helper cell, and the T helper cell releases cytokines, and it activates the B cell into its terminally differentiated form. That is turning the B cell into something called a plasma cell. And the plasma cell is what's going to secrete the antibody, which again is effectively the B cell receptor, which loses its transmembrane region and then generates the antibody. So I want to take a moment to just mention that something that we haven't talked about, which is this concept that, and I've kind of hinted at it, but I just want to make sure that we're on the same page. When you get T cells and B cells out of the thymus and the bone marrow, what you're getting is a subclass or a already predetermined type of cell. So when T cells come out of the thymus, they already have either CD4 or CD8. They're not both, they're or CD4 or CD8, and they also have a T cell receptor. And that T cell receptor is already predeterminate of what that T cell can recognize. And same thing with the B cell. When the B cell comes out, it has a B cell receptor, and that B cell receptor is specific to whatever it can recognize. So this is not a dendritic cell, MHC, and then it comes to this population of T cells and says, okay, is it CD8? Does it have CD8 and CD4? And that the T cell says, okay, you're expressing MHC class 2, so I'm going to eliminate CD8, and then I'm going to adapt my T cell receptor to recognize whatever's on here. Rather, there's a population of T cells. Well, actually, let me put it a different way. So let's say you have a population of T cells like this, and they all pre-recognize a different epitope, a different antigen. And so when the dendritic cell comes and is expressing this particular MHC molecule with this particular antigen or this particular epitope, it is that T cell that recognizes it, and all the other ones are not involved. They're not eliminated, they're just not involved. And same thing with B cells. So all of the selectability that you have in T and B cells pre-exists. And then so what the macrophage or the dendritic cell is doing is selecting for the one that recognizes it, rather than the T cell truly adapting to what the infection is. You're adapting to the infection by selection. All right. So there are 5 different subtypes of antibodies. They all do very different things, and they all exist in different places. And they all have the moniker Ig because, again, they are from the immunoglobulin superfamily. And so the constant region of all the antibodies, again, they look like this. And so all the different constant regions, as you can see, have different glycosylation patterns, and their glycosylation pattern and segment number of their FC region, of their constant region, has to do with what receptor can bind them and trigger whatever it's going to trigger. And we'll talk about functions of antibodies in a moment. So the 5 types are gamma, mu, delta, alpha, and epsilon, which gives rise to the antibodies IgG. IgG is the antibody that you find in blood, and so it is responsible for blood protection, blood-based transcytosis into the blood-brain barrier, all this other stuff. So it really protects the blood system, and that's where you find IgG. IgM, which is the strongest stimulator of classical complement, and typically IgG acts alone. IgM is made in a pentamer, so that means that 5 of these will actually stick together and form kind of like what looks like a snowflake. There's IgD, which is a very rare and not really well studied or understood type of antibody, but it exists. So, we'll just mention that it exists. Then we have the last 2, IgA, which is found in all your mucus and mucosal linings. So mucosal lining. So anywhere you have mucus, it has the highest concentration of IgA. And so this is by far the most abundant antibody subclass in the body because you have a lot of different mucous membranes. And then IgE, which is a very specialized kind of antibody. This is important for the activation of mast cells and eosinophils. So it is predominantly only produced during a anti-parasitic, and by parasitic I mean anti-multicellular parasite or allergic reaction reactions is when you get IgE. So what are these different subclasses? What are these different types of antibodies? Well, all antibodies have the same capacity to do these 6 different modes of action, except it's 5 of the 6, because 1, antibody-mediated cytotoxicity, is very much a function of IgE. By and large. There are some exceptions to that rule, but again, by and large, this is IgE mediated functionality, but this is an eosinophil, and it's releasing granules to kill a helminth, as you see here. It's a worm. So what are the other five, and what do they do as far as functionality? Well, we're going to talk at this one here first. Opsonization. As you might imagine, as I mentioned before, here are antibodies that are bound. The constant region is effacing, which means that there are receptors on different phagocytes that can enhance the ability for phagocytosis and recognition. And again, this is because your adaptive immune system, right? Your adaptive immune system only has the capacity, really, to kill human cells, right? It only has capacity to kill human cells. So if your adaptive system only has a capacity for killing human cells, it means if you are activating your adaptive system for the purposes of adapting or having enhanced special function, opsonization is a really important one, especially with immunology or immunity against microorganisms, because you have a microbiota. So in this case, we actually can discern, or it helps things like neutrophils and macrophages discern what is a good bacteria and what is a bad bacteria. Because before the activation of the adaptive immune system, they will kill all microbes, right? So a microbe equals death. However, once you have an antibody bound to a microbe, like so, microbe plus antibody is much more greatly sought after for death versus those that are not. And so we can really narrow our contingent inflammation, our sentinel immunity, in order to specifically target the pathogen that's causing the kind of destruction that we're trying to alleviate. Now, is the system perfect? No. Do you still get collateral damage or still get off chance death of bacteria that are not bound by antibodies? Sure, because as much as all the phagocytes are going to produce IgE, IgM, IgG, IgA receptor, they still have their pattern recognition receptors, right? So they're going to have the pattern recognition receptors and IgG receptor, IgA receptor, and IgM receptor, right? So we're still going to have both general and specific enhancement of phagocytosis. So opsonization is a really important function of antibodies. There's another one called agglutination. So agglutination is the ability for antibodies to bring what would relatively be a dispersed populace of bacteria or microorganisms to one specific location. And because they're bound by antibodies, again, they can be found by receptors. And so this phagocyte can increase its phagocytosis of multiple bacteria, but also by agglutinating them, it also prevents them from swimming away. It also prevents them from colonizing other sites. So it helps enhance their stickiness. Agglutination also does help as a function of opsonization, even in the absence of complete coding we see with opsonization. And again, like this is just a numbers game, right? The more antibodies that are bound, the more receptors that can recognize those antibodies, the more phagocytosis that can occur because of that engagement. But again, if you don't have enough antibodies, or the antibody maybe not a really good sticking kind of antibody, agglutination brings all of the, or clumps all the bacteria or all the microorganisms into a similar spot. The next is neutralization. Neutralization is similar to phagocytosis, opsonization, in that there's complete coating or covering of the either antigen or microorganism. And the purposes of this are if I have a bacteria, right, and that bacteria has a receptor, and that receptor is really important for a ligand being produced by a eukaryote. If I stick an antibody here in the way, then it blocks or inhibits the ability for this receptor to bind to that ligand, and so it neutralizes the ability for it to act on it. So neutralization can prevent docking or adherence, which again is a really critical part of the pathogenic strategy, but also really importantly can also inhibit or block the ability of effector molecules such as toxins, toxoids, poisons from binding their receptor targets and causing cellular damage. Activation of complement, all antibodies can do this. The strongest one is IgM, but IgG and IgA have also been shown to induce complement. Again, this is through the classic pathway because it requires antibodies, which again is why classic complementation occurs last. And then finally, it can cause inflammation because antibodies can, in fact, bind to molecules, or they can bind to each other, forming what they call an immune complex. So an immune complex. And the immune complexes actually trigger basically a disruption of cellular flow or cellular metabolism, or basically there's some disruption within a vessel or someplace where things are supposed to move through, and that blockage attracts different kinds of immune cells to hunt for that immune complex and allows for an increase in relative sentinel immunity. All right, so when we have these different kinds of cells and different kinds of activations, again, when we have first exposure to antigens, they're phagocytized and displayed by DCs. DCs activate T helper cells. The T helper cells and activation of DCs to cytotoxic T cells all align for the activation of cytotoxic T cells if it is MHC class 1 in this case. So this, if you're thinking about it too, this means that in this case, if you're doing this and activating cytotoxic T cells, you are having dendritic cells which produce MHC class 1 and at the same time produce MHC class 2, and they can activate both of these at the same time, just like you see in this picture, right? So again, they're expressing class 2 here, class 1 here, and they're activating both CD4 positive and CD8 positive T cells, but again, with a CD to MHC specific activation complex. When this occurs, this subclass of population actually activates, because again, through cytokines, we're going to activate the JAK-STAT pathway, and that JAK-STAT pathway is going to give rise to immunological function and create a population of active cytotoxic T cells and active T helper cells. But we're also going to generate something called memory. And these memory cells do not leave the lymph node. They actually, during the course of infection, will stay in the lymphatic system. And the active cells, so the active cells here, will go and leave the lymph node. They'll leave the lymph node through the efferent So we came in through the afferent, we're leaving through the efferent lymph. In order, and then also we can be either leaving through some form of artery or venule system. So we can jump right into the bloodstream, or we can get into the lymphatic system. And so these cells then leave, go fight infection. And then once the fighting is done, memory T cells actually will leave, they'll vacate the lymph node, and they will go to the tissue that was infected. And this is where they will reside in the same tissue that was affected for lifelong, or at least, depending on the generation of memory, a multitude of years, if not lifelong, uh, residence in the lamina propria of those tissues. So as I said before, macrophages and dendritic cells are always in all of the different tissues, but they're not the only ones that reside there. And this is one example of the types of cells that live in certain tissues that have been infected by a certain kind of microorganism. B cells also give rise to generate memory B cells, but memory B cells remain in the lymphatic system. So they never leave the lymph nodes. And same thing with the plasma cells that they generate. And the plasma cells are the ones that are going to secrete the antibodies that help defend us against the certain kind of microorganism. So in some essence, again, we are always getting activation, and it's really important to understand this, that memory cells and effector cells, right, or plasma cells and memory and B cells are generated in order to basically enhance our ability for secondary exposure to a certain pathogen. So the idea is that if something has caused an infection before and you have the correct selection of those T and B cells, by having them in the tissue and having them reside in that common lymph node to that infection, leads to an increased expedience and a more potent response because of the quickness in response, because they don't have to relegate themselves by being activated and still going through the selection process. They already exist and they've already selected, so they've already clonally proliferated, right? Because again, if we're doing this JAK-STAT pathway and we're giving rise to our different effector cells, like such, these effector cells are again increasing in protein production because they're activating their immune specific proteins through activation of NF-kappa B, and they can do so because the JAK-STAT pathway increases AKT signaling, which is important for protein synthesis, and cyclins, which is important for cell cycle. Now, I want to take a moment before we go any further. And so, we have this idea, we have this concept of what B cells do and what cytotoxic T cells do, right? Cytotoxic T cells, they're going to engage with MHCs, and just like natural killer cells. In this case, it's the duality. NK cells are looking for the lack of MHC molecules. T cells are looking for specific MHC molecules that have a certain epitope on them, and if they do, this will kill all the cells that bear that particular marker that activated that particular T cell. So I want to share with you really quickly the different kinds of T helper cells. So with T helper cells, we have 4 really important canonical responses. They are Th1, Th2, Th17, and Tregs. And I'm going to go through just what these cells secrete and why they're important. So Th1s, these secrete IL-12, IL-1, and TNF alpha, and interferon gamma. And as such, these increase macrophage M1s and increase NK and cytotoxic T cell function. So effectively what Th1s are for and what they're activated for is your antiviral, anti-tumor, anything that increases the ability or need to increase human cell killing. And they also increase M1 macrophage production because those are the hyperphagocytic cells. So if you're killing a lot of cells, right, if you're killing a lot of cells, the cell debris is then destroyed or eaten by these M1 macrophages. Th2s secrete IL-4 and IL-5. Again, this is canonical for the activation or expression of IgE and activation of your mast cells and eosinophils. And so these are, or Th2 is typically important for extracellular, what we call phogons. And so you can have some Th2 when it comes to bacteria, fungi, but predominantly it is for protists, both single cell and multicellular protists. Th17 secretes IL-17 and IL-8 and increases IL-6 production. And this enhances neutrophils. So it causes higher activation and cell division. So you're increasing the number and activation of neutrophils. And then finally, as we talked about, the circuits of immunology, right? Both complement, cytokines, this constant circuitry of inflammation has no stoppage in a vacuum. And this is where Tregs come in. So regulatory T cells or Tregs, these secrete tumor growth factor beta or TGF beta, and most importantly, IL-10. And IL-10 and TGF beta set to resolve or remediate inflammation. So when these come into town or into the tissue that's inflamed, the secretion of these molecules help reduce or retract all different kinds of inflammatory cells. So it is an essential component of tissue protection post-inflammation in order to remediate or resolve that inflammation. And these are the four. Now there are others, but these are the four major types of T helper cell responses. All right. So moving forward, right? So as I mentioned before, we're generating these memory responses. We're generating them again at the same time, right? We're generating them at the same time or during the response that we're getting. And this results in something called immunological memory or protective immunity. So when we are first exposed to any particular antigen or pathogen, it takes anywhere between, again, look at the time scale here, 7 days, 10 days, and we eventually get a production of T cells and B cells. But again, there is some level of hesitancy and some level of lack of potency simply due to the fact that it takes a lot of energy to generate this response. And again, the body is already expending a lot of energy, because if you're sick for 7 days, I mean, you probably can understand this, right? If you've been sick for 7 days, you have been battling with an infectious agent. You're spending a lot of energy creating and allowing for the function of neutrophils and macrophages and all these other cell types. And so the amount of energy that is expended over those 7 days in comparison to what you have left in stores to create these T and B cell responses is far limited, just based on sheer kinetics, based on sheer amount of time. However, either by repeated exposure or not successful infections, you can get years upon years of relative protective response or release of antibodies. Again, so here's your antibody curve. So your antibodies last for years and years and years and years. And now when you're, again, infected, not exposed to, but infected by the same microorganism, it's kind of hard to tell because the change in dynamics here. But when you get infected, this 7 to 10 day time frame is completely shortened to about 24 hours. So within 24 hours, where you have not spent a ton of energy and you haven't expended 7 days of energy fighting off an infection, you immediately get activation of the same B cells, those memory B cells and those memory T cells, in order to make new plasma cells and effector T cells that have a much higher, right? They have a much higher and much more specific response to the second infection. And this has a lot to do with not only the selection and the function of memory cells in relationship to the potency that they can generate, but also think about it like this. I told you that macrophages and dendritic cells can activate T cells by being professional antigen presenting cells. Well, now, if memory T cells and memory plasma cells are in the very local, the common lymph node local to that tissue, and the memory T cells are in the actual tissues, if pathogen interacts with the macrophage expresses on MHC, and the T cell exists right here where they can now just respond. They don't even have to migrate. They don't need to extravasate. They don't have to do anything. They just start clonally proliferating and then they respond. This leads to this massive enhancement of potency and kinetic timing, and going from 7 to 10 days to 24 hours. And when you do this, the second exposure, or in the case of vaccines, such as a booster shot, you get a much more drawn out protective immunity response. So you get this massive and much lower curve rate of your antibody production, which remains protective over time. So just as a component of this, right? If we're looking at this from the perspective of virus specific antibodies, and this is a case study in for smallpox. So what they found is that virus specific antibodies are very, very little in people who are unvaccinated, but they're tenfold higher even 80 years after inoculation. And same thing with your T cell specific memory. So this is antibodies. These are T cells that can come out of populace again. 75 years after vaccination, still have 60% of your 60% of the individuals have those CD8 positive T cells, which can kill those cells that are infected with the virus. And even that same 60%, same thing, 75 years after, are still producing antibodies against um, against these cells. So there's something to be said about this concept of, well, okay, if that's true, and you can go 80 years without knowing this, like without having any dip in immunological memory, then why do we need a booster? Well, you need boosters because, as you can see, as you get older, right, in the 50 years since vaccination, you do start to get a dip in protective immunity. Same thing here. Um, you start getting a dip in um, in more protective immunity. And so what boosters do is, as you get some like dips, because again, you do require some level of repeated exposure, and this goes to just some fundamental dynamics of the immune system. Um, and this is also in the absence of mutagenesis of that particular microorganisms, like the flu. Uh, the more mutagenic a microorganism is, the less effective most vaccines are, or at least as of right now, because we haven't found the right components of that particular organism, and can't really target. We can't really figure out the model by which those things mutate in a way that still allows the organism to be successful but escape vaccine immunity. So this is just the current vaccination schedule provided through most physicians' offices that deal in pediatrics. Um, as you can see, you get great number of different boosters for different things like Tdap or DTaP, polio, um, MMR, and all of these different things, influenza, rotavirus, so on and so forth. And only a few of them, meningococci and human papillomavirus, are given, at ages where you might be more exposed to those, like if you're living in dormitories, or if you're engaging in sexual activity, where you might be more exposed to things like papillomavirus or meningococci. And so these aren't typically given to children, not to say that children can't get meningitis or papillomavirus through vertical transmission and the like, but there's a significantly less likelihood that they do. Um, Now, a lot of these different vaccines that we have shown to have really good efficacy, either in the way of um, completely preventing infection. And again, this goes directly to the mutagenesis of an organism. So the more mutagenic um, a particular microorganism is, the less I don't want to say effective, but I would say the less effective the vaccine is in preventing, in complete prevention of infection. So in the case of polio, where polio is very, very non-mutagenic, we were able to generate two different vaccines. And again, if you look at reported cases per 100,000, we were in 30, almost 40. There's a spike in 1952 of a polio epidemic, and so you were getting 35 cases per 100,000. So you're talking really about tens of millions of people who were infected with polio. And when we finally came up with a vaccine, those numbers completely plummeted. They fell off a precipice. And then when the second vaccine came into production, polio was more or less basically not seen again in the United States, and this is for the United States, since basically the mid 1970s. And I think it has effectively been eradicated in most industrialized nations, and only is really endemic in the far the far southeast. So places like the Philippines, East India, places like this, you can still find it. But it really goes to show you that when you have vaccines, at least what the data say is that when vaccines are available for something that is again not mutagenic, and you get a lot of compliance in people receiving that vaccine, and that again, so the more people who have the vaccine, the more prevention you get, it directly correlates to lowering mutagenesis, because again, the fewer people infected, the less mutagenic. And so when that occurs, you get these large drop offs of infection or effectiveness of infection, because of the ability of generating again that shortened immunological response. Because you get, again, if you think about it, if you're giving the first exposure as a vaccine, right, when you actually get infection, this is the response I get and not this. So there is this concept now known as reverse vaccinology, where people are trying to find components of the surface of microorganisms. Because again, if you're trying to develop an antibody, for example, antibody's going to face the membrane, stick out of the membrane. So you want to consider things that are in the membrane. When we think of these really important components of we think of these really important components of bacterial adherence, bacterial pathogenesis, microorganism functionality for infection, we can take advantage of this knowledge in today's reverse genetics world. And so when we find these molecules and we test them and we determine them to be an important component of that microorganism's pathogenic strategy, if we design antibodies to these specific things, it then allows us to then take away the entire organism and just give this protein and this protein and this protein as a vaccine. And so you develop antibodies to these specific proteins. And if they're really important for the function of that microorganism, you're basically either going to cause opsonization, neutralization, or agglutination. And one such example is Tdap vaccine. So the Tdap stands for diphtheria, tetanus, and AP stands for acellular pertussis. So all this is actual, bacterial components, and rather than whole dead microorganisms. And we can look at these things that we have vaccines for, and the relative rates of infections. Yeah, when was the last time since we're not doing this in class, think to yourself, when was the last time you heard of a diphtheria outbreak or tetanus outbreak or even a pertussis outbreak, or typhoid, fever, plague? These are things that cause massive epidemics, even global pandemics back in the classical world and even beyond, well into the Middle Ages and even well into the 1700s, 1800s, like cholera. And since we have developed these vaccines, you've not really heard of these outbreaks except for places where the vaccine's not available, i.e. the non-industrialized world. And same thing with these things. When people stopped taking these vaccines, we started getting signs for people that were warning people of getting their MMR boosters, because people were starting to hear about measles making a return or mumps making a return. But again, have any of you heard of a yellow fever outbreak, which killed a lot of people when the Panama Canal was being created? Or polio, again, basically eradicated from the United States since the mid-70s. Again, the influenza virus. This is not, again, for prevention of infection, for the virus. So for things that are highly mutagenic, for things that we cannot create really good vaccines for, if it's not for prevention of infection, it is for the limiting or the reduction of burden. So as an example, most people who get the flu when they're not vaccinated for the flu can be sick upwards of 10 to 14 days. Whereas most people who get the flu who are vaccinated for it typically might be sick for a couple days. And that change in kinetics is because of the change in kinetics of the activation of different components of your immune system. Um, Now, comparing them to things that we don't have vaccines for, such as malaria, schistosomiasis, TB, diarrheal diseases. Diarrheal diseases is a big one. In combination, cause almost 2 billion infections a year. That is, for those of you playing the home game, is 1/4 of the global world populace. We all have probably heard of HIV/AIDS. 35.3 million people. An average of another 2 million per year and die of these particular diseases. Same thing with diarrheal diseases. Um, and so what's the take home here? Well, we have vaccines here, and a lot of them you probably couldn't tell me the last time you had an outbreak of these particular microorganisms, except for the flu, but we get the seasonal flu. But how many of you are aware that we have these big problems with things like malaria or TB or AIDS, or think of like foodborne illness and diarrheal diseases? And that the answer is, well, yeah, I've heard of these things, and they're not hard to find in human society. That's why, because we don't have vaccines for them. So one last thing about vaccines really quickly is you might be thinking to yourself, well, Dr. Morrell, you said to us before that the immune system only really works if in fact there's damage, right? So complement requires damage. The activation of basically of the entire immune cascade requires damage, but vaccines don't cause damage. You're just getting a shot, and they have these components in them, but there's no such thing as damage. Well, this is where a really important thing known as an adjuvant comes into play. So adjuvants are created, and here's a list of some of them, and I'm not going to ask you what any of these are. But the idea is that the adjuvants agonize different components, especially like TLRs, in order to stimulate the idea that there is damage. So they induce a state of inflammation in the absence of actual damage, because there's no vaccine that no one's ever going to give you that is ever going to make you sick, unless you were allergic to the components of the vaccine. Then that could make you sick. Now, are all the vaccines great? Can some of them have some side effects? Of course. I'm not going to sit here and tell you that all vaccines are perfect, because some of them will generate a response, and a lot of that response is a combination of the components of the microorganisms that you see here, the different components that your body's going to respond to, because you are generating an immune response, because you're simulating that there is some type of damage and agonizing the receptors specific for the generation of an immune response. And all of this has to do with this concept of both personal generation of memory, but also really important for this concept of herd immunity, because not everybody can generate a vaccine, or not everybody can have a vaccine. Like people who lack. So again, if T cells are required for activating B cells, and these are both required for specification of immunity, what if you don't have T cells or B cells? And there are people who exist like this. And those are people who are considered immunodeficient. And if you're immunodeficient, especially in these kinds of immunodeficiencies, well, you're not going to be able to generate immunity based on vaccines. But the concept here is that if there are people that are completely surrounded by people who are vaccinated, then the infectious people, basically by way of interacting with all these different people, can't generate an outbreak because everybody else prevents infection. So I'm going to leave it there. We can come back to the rest of this when we come back to class, and I hope you guys enjoy.